good afternoon, and once again, very warm welcome to all today's webinar. Thank you for taking time out and being here today. This is Jyoti Negi, Assistant Professor at Siddhartha Institute of Pharmacy. It's a great pleasure for me to introduce our speaker today, who is going to talk to us about Pharma Revolution 4.0 and its effect. Now, I would like to introduce today's speaker, Dr. Mohammed TahirAansari, Associate Professor at School of Pharmacy, University of Nottingham, Malaysia. Now, sir, I would like to invite you to share your knowledge with us. Over to you, sir.
Thanks, Farouk. So let me convey my thanks to the organizers of Siddharth Institute of Pharmacy for inviting me to present a lecture on the concepts. What exactly is happening in the industry at the moment, and what exactly we are doing now, and what is going to happen in future. So how exactly we are going to expand, and how it is going to hit us in terms of availability and manpower.
Right, sir. It's our pleasure
Yeah,
for
thanks a lot.
accepting our invitation.
Yeah. So let me begin with you just to look into the past, what exactly we have done. And what the industry have done is like it started with the revolution concept, which we call it as IA 1.0. We can call it as Pharma 1.0. And I've taken example of tablet press, which was very, very primitive with a single punch. And we used to get a single tablet. So there were some rapid developments, and then we started getting multiple tablets from the same machine with increased number of tablets, and it was actually electronically controlled with the help of pump. With the third revolution, what exactly happened is like it become more and more sophisticated, and it was further classified as a unit operation, still working as a batch process, probably can be converted into a continuous process, which will be a reality in IR4 or Pharma 4, since we are into the aspects of involving machine learning and Internet of Things. So just have a look exactly how it is going to change. Things are going to change. Now, for example, we're using basic statistics like mean, median, mode, student test, and chi-square test and all. With machine learning, we use different concepts. So possibly, things are going to move at a very fast pace. And if we would like to be much more relevant for the department, for the industry, then we need to work hard and change. So I'm not going to say that things are going to change tomorrow. It will take time, and we still have time because whatever things you are looking at, machine learning, it started in the year 1960. So it has taken a long pace for them to be relevant to the market that is 2020 now. So it's almost like 50 years for them to become relevant. So probably we also need to manage ourselves so that we are relevant to the industry. So things are changing and changing for good, but changing at a rapid pace. So we also need to change. So one of the concept is, what exactly the industry is looking at is applying the concept of Six Sigma. And this Six Sigma is actually allowing the industries to work better because it will actually remove the waste. It will improve the quality, and probably it involves the process also becomes much better. For example, we used to do multi-step synthesis previously. Now this has been reduced because of high throughput screening system. And now we have like much shorter synthesis process for tablets. The same happened. It used to be like it take 3 hours, 4 hours for it to be dissolved. Now we have multiple, melt in mouth dissolving tablets and so many things. So Six Sigma is actually gaining an importance, and almost every industry is trying to adapt to the concept of Six Sigma. So unless until we know what exactly Six Sigma is, so we won't be very much relevant to the industry. So Six Sigma works in a way that it will actually channelize the process in a much better way, so that we know exactly what we are doing, we know exactly how we are doing, and we know exactly how to improve the process. So for us, when we speak in terms of products, we speak in terms of new products, and we speak in terms of generic products, which is called as existing. So the channel would be slightly different because here with the new products, we have to actually verify those systems, and with the generic products, we just need to improve and control the system so it is working, if not better, at least as comparable to the existing compounds. So this is just a layout exactly what we do in the Lean Six Sigma approaches. So the first is we need to define the process, measure, analyze, design exactly how to improve all those process variables and material attributes we have identified, and then we need to verify. With if we have a generic product, so probably we try to improve the process and also in a sense that control so that it is within the limit what exactly the innovator has the qualification strategies. So one of the things what exactly in the industry is doing is quality by design. It's still done with the help of software, but a lot of manual aspects are involved. And these all manual aspects are going to be taken off with the application of new systems, which are almost developed, are already in the market. Probably this year there was a competition and they've won an award. So I'll just give you a hint exactly what the product is called nowadays. So just to give a hint exactly, quality by design is a very systemic approach for anything we need to develop. So we know exactly. So there are a few processes what exactly we need to follow. So the process is already written, and we know exactly what to do, how to do, and how much to do. So this will actually pace our production process in a much better way. So not only for the production, but also for the research. We do use it for research, but probably when we speak in terms of IR4, probably, of course, it relates to production and analytical in the industries. So quality by design, the good thing when we adapt the attributes of quality by design, we are able to address the needs of ICH guidelines, which is probably Q8. So whenever we do an industry, we have a lot of paper values and paperworks. So probably we are able to address some of the ICH guidelines with the help of quality by design, which starts from defining, determining, analyzing, designing, controlling, and further improving the process. So with defining exactly what we need to define is we need to define the therapeutic profile based on exactly what the customer wants. So the customer is a physician, is the pharmacist, and the patient. So looking at the outcome, what exactly the customer wants, we will define the therapeutic equivalence, the pharmaceutical equivalence, and the bioequivalence of the product. We will choose what are the quality attribute which will affect all these therapeutic equivalence, pharmaceutical equivalence, and bioequivalence of the product. We will also see exactly how to identify how much a particular attribute will be, posing a risk, and if it is posing a risk, how to analyze, and if we are able to analyze, how to evaluate, how to interpret that analysis. And then when we are able to define all the material attributes and the process parameters, probably we'll look into designing a particular product. And for that, we need to have a particular way exactly what to do, which is statistically verified and statistically validated. So we call it as a concept of design of experiment. So I think most of us would be knowing because we do use this concept a lot in formulation and development. So once we are able to screen and optimize the process, that product is supposed to be verified and controlled. So most of the time, what exactly the industry does is they will run few batches of the same product, like maybe 3 times or maybe 5 times, to get the similar results with all these definitions and the how the screening and optimization is supposed to be done, just to verify exactly the process what exactly they have developed is actually controlled and can be verified. And finally, we can think of exactly how it is going to be used in the industry when we actually advance it in terms of piloting. So quality by design is a wide concept. It can be used in analytical methods, in manufacturing, quality assurance, regulatories, formulation and development. Of course, like F&D and AR&D is the area where exactly most of the academic people are working in, but only applying the concept of DOE. So QBD is having like 6 aspects. So I'm just covering 4 of it. The fifth and sixth is more industrial. So as I said before, the QTPP is supposed to be designed on the basis of what are the customers. So the customers would be internal, which are formulator, manufacturer, and QA, because they are the one who will be deciding how to do. But what to do will be decided by pharmacist, physician, and patient in terms of pharmaceutical equivalence, bioequivalence, and acceptability and compliance. So this is very, very important. Acceptability and compliance can overrule when we don't have, the product is not bioequivalent and pharmaceutical equivalent. So we need to look into the detail exactly how the patient is accepting it. So sometimes, we need to have a balance exactly so that we can, and this balance is actually maintained by all these internal customers. Okay, so what are the quality drivers exactly when we intend to have a quality target product profile? So the quality drivers are these, and driving these, what are the micro factors? So if we speak in terms of tablets, so probably it will be, the assay, the drug uniformity, the impurities, the hardness, the disintegration, the residual. So all these microprocess will be affecting the pharmaceutical equivalence, and probably this will be affecting the customer need. Similarly, with the drug performance, the pharmacokinetic process will be affecting the bioequivalence, and of course will be affecting the requirements of all these 3 PPs. Okay. And storage, which is of course will be also affecting the acceptance, and further it will be affecting the outcome, what exactly the pharmacist, physician, and patient needs. So these 3P are very, very important for us to design any particular product. So this is an example of a tablet. So this is the way exactly how the QTPP is supposed to be defined. What is the target, and why? What are the factors which has been taken into account, and why we have taken into account? So we need to have justification for each and every process. We would be looking into, studying into detail. So why exactly we have done so? So we need to have a justification. Once we are able to design that, so probably we need to confirm that in terms of drug substance characterization, in terms of reference drug product characterization, and in terms of reference drug product packing labeling. So these are some of the industrial aspects which normally every industry now is complying with. So these are the tables which actually are from the industry, so are complying with, and these tables refers to the manufacturing of tablets. So those maybe who have done tablets in industry, they may be familiar with these tables. So the second aspect for QBD is the product management. So it actually allows us to manage the production in a much better way. So the first is we can have a work breakdown structure. So the work breakdown structure is already defined, and then once we are able to define this, we can have a milestone. Okay, and once we have a milestone, so probably we should be able to know exactly who is the responsible person for addressing this milestone. So at the moment, all these things are what the industry is doing is still manual. So these are going to be automized very, very soon, maybe if not next year, maybe 2 years or 3 years down the lane, because the product is already approved by FDA, US FDA, and so probably I think they are going to release it. So once we enter into the production cycle, so we need to, this is the same slide. So we need to know exactly which are the things we need to qualify it as quality attributes. What are the things which should be qualified as process parameters. So we need to address this by analyzing the particular aspects through impact analysis and severity analysis. So it says that any changes in the formulation will actually affect the quality assurance or the quality parameters of the drug. If yes, then probably it should be classified as it should undergo severity analysis. And if it passes the severity analysis, only then it is supposed to be qualified as quality attribute. So these two are very, very important for us to qualify which particular aspect is supposed to be classified as critical quality attribute. So this is again an example. So these are the quality attributes which have gone through the aspects of impact analysis and severity analysis, and these are the one which is supposed to be critical for finished product. Similarly, these are the quality attributes have gone through the checks of impact analysis and severity analysis, and these are the quality attributes, critical quality attributes for in process for tablet designing. Okay. So once we are able to identify the risk, we can have a structured way exactly in which we can show it. So there are two ways. Either we have a process map, a fishbone diagram. So nowadays, since Six Sigma is involved, so we use a lot of fishbone diagram. The second aspect is analyzing risk. I mean, how much it is risk, those quality attributes, what exactly we have filtered, how much it is risky for the process, and exactly how we can claim that these are risky. So these are the three process: identification, analysis, and evaluation of those segregated quality attributes. So this is an example again of a risk assessment done by fishbone diagram. So probably it's easy, one chart will be able to define all those risk factors which are involved. Once we are done, we will use either two methods, either we call it as relative risk matrix analysis, which will classify the risk factors into high, medium, and low. So those factors which are supposed to be classified as high will be taken into account, will be further analyzed, and will check exactly if these processes are supposed to be evaluated or not. Okay. So the second method is by failure mode and effect analysis method. So this also will help us to access the risk analysis. And once we are able to access, we will assign a risk probability number. So that risk probability number is the evaluation strategy, which will allow us to know exactly if this particular aspects or attributes or the process parameter should be studied via DOE process. So for that, what exactly we do is we assign some the number of occurrence it will be, it is supposed to have. For example, every 5 times we run, solid state of form is supposed to affect the quality attribute, any of the quality attributes such as drug release or maybe drug uniformity. So probably we will keep on assigning the number. So based on the number, RPN number, risk probability number, we will see exactly which of these factors it should be picked into the next phase. So once we are able to design this and come to a particular session, so any risk factor having a probability number of more than 25 is said to be qualified. And these qualified factors should be screened further by DOE process. So for example, here the solid state is supposed to be qualified for the further analysis. Flow properties, which may be classified as critical quality attribute, but still doesn't qualify for the DOE studies or further process because the incidence of flow properties affecting the the quality of the product is less. The same goes for, this is for API, this is for excipients and for the process. So we have to do a risk analysis for API, for excipients, as well as process. Okay, now comes the next step, which is DOE. So most of us, we know exactly what is DOE. So DOE is the one which will study the relationship between the input factors and the output factors. And this actually will decide how much these factors are going to affect the properties which are further going to affect PPP, patient, physicians, and the pharmacist. So these are some of the rules when we apply DOE. So I'm not going into detail because DOE is a quite vast concept. So just some of the rules, what exactly we'll look into so that a particular process qualifies for the next step. So these are some of the statistical values which should be taken into consideration, if not all. So probably this will help us to know exactly what are the various, those factors which will be responsible for us to have an optimized formulation. Okay, so there are many DOE process, but normally we classify DOE into screening and then optimization process. Screening is done if we have more than the required factors, and we cannot study all those factors for the optimization purpose. So we screen which may be responsible for the optimization process. For screening also, depending on the number of factors, we decide, for example, if we have more than 6, we use placket Berman. If we have 4 to 6 factors, we'll use fractional factorial design. And if we have less than 3, so we have 2-level full factorial design. So this is the screening process. Once we are, if we don't need to undergo a screening process, then we have limited factors, and all factors are going to affect the final product of the, the final, whatever the result is. So then we can go for optimization process. So this is like 3-level full factorial. It can be response surface design, or it can be a mixture, particularly if we have a fixed ratio of the components. So this is only when we have a fixed ratio of the components. Otherwise, most of the time we'll be using full factorial or response surface design. Under response surface design, we have just to have a look like box Penken and the CCD. So these are the 2 major response surface design. So I can't go into detail because it's quite a long thing to be studied. Okay, so with that, I'll just share you the case studies for lipid nanoparticles. What exactly we did is like we came up with these as the material attributes. So these are the possible material attributes which may affect the final results of the nanoparticles in terms of size, in terms of resolution, in terms of assays, in terms of loading. So, and along with that, the material attributes, these we saw that most of the cases, the speed, the time, the homogenization pressure, the sonication amplitude are the one which will be affecting all these attributes. So this is a case exactly we can use all these things to carry out a DOE study for any lipid nanoparticles. For transdermal system, so you can see like the material attributes further increases because there are a lot of excipients issues whenever we do a transdermal system. Because most of the time the transdermal system, the bases will be hydrophobic, the drug may be hydrophilic, so these kind of things occur. So we came across these as some of the material attributes which should be taken into account, and these are the process parameters. So depending exactly, I don't say that we need to consider all, but probably we need to screen this. So if the number of CMAs and CPPs are quite high, so probably we need to have a screening system before we end up in the optimization process. So this is what exactly we are doing at the moment. So just to share exactly, we are doing lipid-based nanoparticles. So probably, so this is what exactly we got some of the results. And to end up, I would say that what exactly the pharmacopoeia is doing, even majority of the drug regulating agencies in each and industries are actually hitting it very hard so that all these concepts are actually regularized. So British Pharmacopoeia have taken a step ahead, and they will be including a monograph in the new British Pharmacopoeia 2022. Since we refer BP a lot, so probably we are waiting for that update, and that is actually a chapter which is on analytical quality by design method. So probably once it is included, so it will be mandatory for every industry to have this process. Any analytical method development is supposed to undergo this screening process using the OE method. Now, this is the system which is coming up. So this is called as knowledge aided assessment structured. We call it as CASA. So this CASA is the one which is coming up with which will have all the algorithms. What exactly we have just spoken in. So all the algorithms will be embedded. All those aspects will be embedded as an algorithm, and we as the users have to train those algorithms. So, so we can't train unless and until we don't know. So this will be trained, and the training is supposed to be continuous. It doesn't mean that once we train, it will be on its own. No, even the system, laptop and all, it has improved so much, but still it requires training. So we need to keep on training, and training and training. And this literally will be on its own. So instead of doing all these 6 chapters, I mean, here we can say for all these 6 chapters individually. So this CASA is going to organize all into one. So this is what exactly which is going to happen. So they have already launched it probably, and this year they have won an award in innovative some kind of competition. So probably they will be coming up with the actual software very soon. So this is exactly what I was talking. So they've already won the innovation award in this particular conference. So that's all from me. If you have any questions, you can please, yeah. See, farming, and nowadays you see, is fourth revolution is going to change everything. So it's very vast. What exactly I have shown you is just it's an aspect what exactly how the pharmaceutical industries are going to change. If pharmacy practice are also going to change, have started changing. If you can see, I mean, I don't think so. It's like India is changing at such a large pace, but other countries are already changing. So probably, yeah, it will change.
Okay, sir. Sir, one more question. How industry revolutions affect global economy?
Oh, so definitely every industrial revolution actually have a positive impact on the industries. We can see exactly how uh, the countries have grown from having been nothing and having everything now. So definitely it is going to have a positive effect on the economy. That's one. But probably having said that, it is very important for us because we are the one who will be witnessing the change. So if we are the one who are witnessing the change, so we also should know exactly what is going to change, and we should be the part of that change. Otherwise, it will happen like what happened to Kodak. So when things were changing, Kodak didn't change, and it can't flourish. Same happened with Nokia. When Nokia, everything was changing to Android, and Apple was already on a running spree. Nokia was not able to accept that changes, and probably it has to vanish. So when we are changing, we need to be a part of that change. Yeah.
Okay, sir. Thank you, sir. This lecture certainly benefited from your knowledge and deep of expertise. We have come to the end of our webinar. Now, I would like to call the program convener. This is Mrs. Himani Nautiyal, ma'am.
Thank you so much, ma'am. Good afternoon, one and all. It's been very fascinating and engaging day, so kindly indulge me for more 5 minutes. As on the behalf of Siddhartha Institute of Pharmacy and its entire fraternity, I feel immense pleasure to propose a vote of thanks. Firstly, I want to express my sincere gratitude to Dr. Firoz Anwar, who spared precious time from his busy schedule to grace this occasion and enlighten all of us with his talk. So rightly stays and talk about his topic. Thank you so much, sir. We are truly honored to have you with us. Now, I further extend my heartily thanks to Dr. Farooq for his thought-provoking insights on his topic. Thank you so much, sir, for your acquainted research talk. And we will look forward for your guidance during incorporation of such experimental model in our institution. Thank you, sir. I would further express my gratitude to Dr. Lee for such interesting topic. Your work was truly inspiring and kept us spellbound throughout the session. Thank you, ma'am. I would extend forward my gratitude to Dr. Ansari for sharing your excellent knowledge with us. Thank you so much, sir, for your unique topic with precious time. Now, I would express my humble gratitude to our chairman, Sri Durga Prasad Verma ji, for having faith in us, our director, Dr. Rajiv Sharma ji, for his continuous appreciation, and on the behalf of even committees of this program, I would extend a heartily thank to our mentor, our motivator, and organizing director, Dr. Sanjay Singh, for his continuous encouragement and valuable support. Thank you so much, sir. And it would be incomplete if I won't acknowledge the whole team behind this program, including technical, organizing, registration, and media committee for their dedication and patience in terms of their task. Thank you so much, everyone. Now it's time to wrap up the day. We would thank our participant for their active participation and for sticking with us in this entire session. We look forward for more of such insightful conferences and webinars. Thank you everyone for such a fruitful day and have a wonderful evening. Thank you so much.